Good morning or afternoon or evening, whatever time of day you're watching this. I am going to go over slides 26 through 41, everything we did not finish in our last notes packet. Please get this done as soon as you possibly can because you will need this for this week's practice problems that we're going to be doing on incomplete dominance and codominance. So I would highly recommend you get that done as soon as possible. Alright, with that, this is the notes packet. I made a separate part for this one so you didn't have to continue on the same one that you had, and this one is the one that you will submit, or if you printed out the other one you can do that. I did make some minor changes though, so it might not match perfectly. Sorry, I don't know what else to tell you. Okay, let's look at our notes. We are starting on slide 26, and this is on types of crosses. And we have a dihybrid cross, has a cross of 2 traits. And we did these in our practice problems last week, where we did the running and waltzing mouse with the brown or black fur. And when you do the crosses of these, you get, each parent has 4 possible gametes. So like when we did big R with big B, little b, we remember how we took the first trait of the first one, the first allele of the first trait and combined it with the first allele of the second trait. That is 1 gamete that it produced. So that would be big R, big B. And then we took our second, our first allele with the second allele of the second trait, and that was our second possible gamete. And then we did the same thing for our second allele. So our second big R with the big B, that would be a third possible gamete. And then a fourth possible gamete was the big R with the little b that we talked about. So that would be each of these rows, the ones that are sitting on top was our possible gamete. So parent 1 has 4 possible gametes in this cross, and parent 2 has 4 possible gametes in this cross, which means you have 16 different possibilities for trait combinations for your F1 generation. So that's one way to think about it, is your gametes are on top, the different gamete possibilities, and then how those combine with the both parents coming together, that is your trait combinations. Okay. Slide 27. I went backwards, not forwards. There we go. Other patterns of inheritance. So I've mentioned this a couple times, incomplete dominance, and that is when an individual shows a combination of the inherited alleles. These crosses will involve 3 possible phenotypes or traits. Such like you might have a red flower and a white flower, and that might produce a third phenotype that neither of your parents had, and that could be the pink snapdragon flower. Okay, and then the same thing as if you have a person with straight hair and a person with really curly hair, you might get an offspring with wavy hair, not quite enough to be called curly, and definitely not straight enough to be straight. So we have 3 possible phenotypes as our F1 generation. Alright, so we have some examples. So on your papers, we have big R, that will be red. Big W would be white. A big R, big W would then be pink. That would be that codominance right there. I'm sorry, incomplete dominance. Just kidding, ignore that. Incomplete dominance. We could also do it with black and white. So big B would be black. Big W would be white, and if you get a mixture, you get big B, big W, that will give you the gray type. Okay? Let's cross a red, big R, feathered bird with a yellow, big W, feathered bird, and that will result us in an orange RW feathered bird. And we're going to do this one on here, so let me exit out. There we go. So let's cross it. We cross a P1 cross. Remember, we like to always do that. Is big R with big Y for yellow. Okay. And we put our big R's here and our Y's here. Oh, I guess our P1 cross was supposed to go here. Sorry. I wasn't paying attention. I just modified this 20 minutes ago. You'd think I'd remember. Evidently not. It's too much for me. Okay. Alright. Big R's, bring those down. Okay, and then bring your Y's across. And now we can tell what are our offspring's phenotypes. Well, RW is going to be orange. How many of our offspring are going to be orange? Well, in this case, 4 of them are going to be orange, and none are going to be anything else. Genotypes of our offspring is RY, and our genotype ratio is 4 to 0 because they are all RY. Okay, that one's pretty easy, hopefully. And back to this. Okay. Other patterns of inheritance, besides incomplete dominance, we have codominance. And this is when you have two dominant alleles that are being expressed at the same time. So, for example, roan horses can have both red and white hairs in 50-50 equal amounts. That's because they have both red and white are dominant, and it shares it. And they're both expressed. Another thing that we have is multiple alleles. Notice in the box here, we will be doing a practice worksheet on this, so don't worry, we will get plenty of practice with blood types. So the blood types is a perfect example of multiple alleles because this has genes that have 3 or more alleles that type for it or code for it. So blood type, because we have 3 main blood types, we have A, B, and O. Type A, and we use the letter I for blood type, big I, and then in the superscript, so normally we've been doing subscripts, now we're doing a superscript of a big A, that is going to be equally dominant over type B, which is going to be IB, with the capital B in the superscript. But both of these are dominant over type O, which is going to be your lowercase I. So we actually have 4 types of blood types for this, even though there's only 3 genes. We have A, B, AB, and type O. Okay? Type O, positive, and type O negative make up the majority of blood groups in the US. I actually do not know what my blood type is. My husband is B positive. He just found that out. I don't know. I'll have to look mine up or ask for mine. Okay, so if you know your blood type, I'd be curious to know where you fall in here. Alright, so we will definitely be working more on blood type, but for now let's move on to what polygenic traits are, and these are traits where you have several genes influencing the outcome. So, eye color is controlled by multiple genes. Hair color, skin color, your height, all of those have multiple genes that help decide what the final outcome will be. Okay, which is why when we were looking at the pre-implantation genetic disorders, why they were saying that they could not test for that yet, that maybe down the road we could, but because it's controlled by so many different genes, it's a very complicated process. Okay, sometimes we have environmental conditions that will affect what the phenotype will be. It will also help influence the genetically determined traits. So sometimes rabbits will be different colors based on where they are, and they could be the same species but different colors based on their habitat and range of where they live, which is kind of interesting. Okay, meiosis. Here's where I did change things. So if you printed out the first set of notes, I have definitely changed the notes packet for this. So I've got this picture added in. And meiosis is a process that results in cells with half the normal number of chromosomes, or gametes. And then we have the definition of a gamete. Gametes are your sex cells, and that for males is sperm, and for females that's the egg or ovum. Okay? And if you want to talk about ovum in the plural, because women do typically have 2, it's ova. Okay? We might have more than one ovum released, which would be your egg. Sometimes women will release them from both sides. It's not common, but it does happen, and twins do happen. Okay? Alright, meiosis continued. The purpose of meiosis is to reduce the number of chromosomes in half. So we go from 2n from a normal cell to n, and n number of chromosomes is what we would consider our gametes, and that is to form. So the purpose is also to form gametes, to reduce the number of chromosomes in our gametes, and that makes sense because two gametes have to come together, and if you don't reduce that number of chromosomes, you have too many chromosomes in the fertilized egg. So that's a problem, so we have to make sure we have half the number in each. Okay? So haploid is what n is, and it's the number of chromosomes found in the gametes of an organism. One set of each chromosome, so you get one set from mom and one set from dad. So sperms have one set, and eggs have the other corresponding set. We represent that it's haploid by putting the number n. And for example, human haploid number is 23 because we have 46 chromosomes. So what would be the haploid number of chromosomes for the fruit fly that has a diploid number of 8? So 2n is 8. So n would be half of that, right? Because it's haploid is half of the diploid number, so it would be 4. Okay? Diploid is represented by 2n. And the number of it's the number of homologous chromosomes, so those paired ones. For humans, again, like I mentioned, that's 46. Examples of human diploid cells are any body cells except the sex cells. Those ones, as we just discussed, are haploid. All right, a body cell in an adult fruit fly has 8 chromosomes as shown in the figure. 4 of the chromosomes come from its male parent and 4 come from its female parent. These 2 sets of chromosomes are homologous, and that's what you fill in on the paper there, okay? Meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. They match each other. They're complementary, okay? All right. Here is a diagram. We used to teach you every step. You had to know what was going on in each one of them, and not unfortunately, it's just not what we have to teach anymore. There are 2 rounds of meiosis that we go through. The first round reduces or splits rather the homologous chromosomes into daughter nuclei, and then we take that and we break the homologous pairs into individual ones so that you're getting your n number of chromosomes. There is a video here. I have also included the link on your notes packet. I don't think I want to spend the 5, well, yeah, let's just do it. Gotta make it big.
Many organisms pass their genes to their offspring through sexual
Hold on. I need to move this up so that I can click on it, or we'll put it up here. That way I can click on it without the little thingamajiggy getting in the way.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, Meiosis I, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, Meiosis II, results in 4 unique haploid cells that only have one copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis I. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving two pairs of sister chromatids. Chromosomal material is exchanged between the two pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no two siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis I is called Metaphase I. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is Anaphase I. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis I are Telophase I and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis II. Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In Anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In Telophase II, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis II began with two cells, and each of those cells were split into two cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Okay. Go back to this. So hopefully you were able to watch that and at least get some of the idea of what's going on in meiosis. Now let's look at gametes. Okay, gametes become zygotes after fertilization, and the fertilization is the joining of gametes to form that zygote, and the zygote is diploid. Please remember that. So if you see 2N, it's got to be a zygote or a regular cell that is not a gamete. Not a gamete. Or a sex cell, okay? So, and it shows that they will be fusing together here in this picture for you. All right, gene linkage. This is where each chromosome is actually a group of linked genes, and the chromosomes assort independently. So the chromosomes are going to separate out, not the genes, because sometimes the genes are on specific, there's multiple genes on your chromosomes, and they're not going to be separating by genes, they're going to be just separating by chromosomes, okay? That, the box here says alleles of different genes tend to be inherited together when those genes are located close to each other on the same chromosome, and that has to do with the crossing over. If they're close together, the odds are they won't get separated through crossing over. If they're farther apart, the odds are they might get separated from crossing over, okay? The next two slides you're not going to copy down, but I would like you to watch. And this is about gene mapping. In 1911, Columbia University student Alfred Stortevant wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's location. He reasoned that the farther apart two genes were on a chromosome, the more likely it would be that a crossover event would occur between them. That's because if two genes are close together, then crossover events between them should be rare. If two genes are far apart, then crossovers between them probably would be more common. Okay, that's what he was speculating. By that reasoning, one can use the frequency of crossing over between genes to determine their distance from each other. So assuming that that is true, he decided to look at different things. So he gathered lab data and presented a gene map showing the relative locations of each known gene on one of the Drosophila chromosomes. His method has been used to construct gene maps ever since the discovery. So we think it's true, and it turns out probably if it wasn't true, we would know that from the genome sequencing that we've done. But anyway, if you have maybe black hair and reduced bristles, if you're looking at those two, and brown eyes, if you look and see brown eyes is far away from black bodies and reduced bristles, so it's more likely that you'll have this one be separated from this one. But these two are more likely to be found together. You'll most likely see the black body with reduced bristles. You'll also be more likely to find purple eyes and light eyes than purple eyes with aristolas, which is no bristles on the antenna. Okay, so these things are showing you how, if they're closer together, they're probably going to be stay together on that chromosome. But if you have opposite ends, that's where you run into that probably will not stay together. It may very well split up. Okay. All right, that is the end of that. If you have any questions, please email me. Come see me during pause. Ask during class. Nobody asks me any questions during class. I don't think I have done that great of a job, so I'm really hoping somebody has a question or two that you would like me to elaborate on, especially meiosis, because that's a complicated process, and I know we're not memorizing any of it, but if you have questions, please bring them to class. Okay? I will see you guys on Tuesday and Wednesday. Have a great one.